Our second SoftChalk lesson in Unit 1, Lecture 5 deals with bacterial flagella. Let's start out by looking at our fundamental statements or bullet points. So you see that many bacteria are motile and use flagella to swim through liquid environments. The basal body of a bacterial flagellum functions as a rotary motor. It enables the flagellum to rotate and propel the bacterium through the surrounding fluid. Bacterial flagella appear in several arrangements, each unique to a particular organism. Motility serves to keep bacteria in an optimum environment via taxis. Taxis refers to a motile response to an environmental stimulus, enabling the net movement of bacteria towards some beneficial attractant or away from some harmful repellent. Most bacterial flagella can rotate both clockwise and counterclockwise, enabling them to stop and change direction. The protein flagellin that forms the filament of a bacterial flagella functions as a pathogen-associated molecular pattern, or PAMP, that binds to pattern recognition receptors on a variety of defense cells of the body in order to trigger innate immune defenses. Motility and chemotaxis probably help some intestinal pathogens move through the mucus layer so they can attach to epithelial cells of the mucous membranes and colonize the intestines. Innate immunity is an antigen non-specific defense mechanism that the host uses immediately or within several hours after exposure to a microbe. This is the immunity one is born with and our body's initial response to eliminate microbes and prevent infection. An antigen is a molecular shape that reacts with antigen receptors on lymphocytes to initiate adaptive immune responses. The actual portion or fragment of an antigen that reacts with antibodies and with the receptors on B lymphocytes and T lymphocytes are called epitopes. Adaptive immunity is an antigen-specific body defense mechanism that takes several days to become protective and is designed to remove a specific antigen. This is the immunity we develop throughout our life. Humoral immunity involves the production of antibody molecules in response to an antigen. Cell-mediated immunity involves the production of cytotoxic T lymphocytes, activated macrophages, activated NK cells, and cytokines in response to an antigen. And this helps to remove infected cells and cancer cells that display foreign epitopes. Antibodies made against bacterial flagella can immobilize bacteria and or promote phagocytosis through opsonization. And finally, motility enables some spirochetes to penetrate deeper in tissue and enter the lymphatics and bloodstream, where they can then disseminate to other body sites. So those are our bullet points, and now let's look at our detailed learning objectives for this SoftChalk lesson. You should be able to describe the basic structure of a bacterial flagellum and state its function. State what provides the energy for bacterial flagellar rotation. Define and recognize the following flagellar arrangements: monotrichous, lophotrichous, amphitrichous, paratrichous, and axial filaments. Define taxis. Compare and contrast innate and adaptive immunity. State how bacterial flagella may play a role in the initiation of innate immune defenses. Briefly describe how bacterial flagella and chemotaxis might play a role in pathogenicity of some bacteria. And briefly describe how antibodies made against bacterial flagella can immobilize bacteria and or promote phagocytosis. And our highlighted bacterium for this lesson is the syphilis spirochete, Treponema pallidum. So let's start out by looking at the structure and composition of bacterial flagellum. A bacterial flagellum, singular of flagella, has three basic parts: a filament, a hook, and a basal body. The filament is a rigid helical structure that extends from the cell surface and is composed of a protein called flagellin, which is arranged in helical chains to form a hollow core. During synthesis of the flagellin in the filament, flagellin molecules coming off the ribosomes are transported through the hollow core of the filament, where they attach to the growing tip of the filament, causing it to lengthen. And with few exceptions, the flagellar filament is not surrounded by a sheath, such as a flagella in eukaryotic cells. The second part, the hook, is a flexible coupling between the filament and the basal body. If we look at figure 1, the filament we see here is the actual hair-like structure we see sticking out of the cell, composed of flagellin. That's a helical hollow protein tube. That connects to a flexible coupling called a hook. And then the hook attaches to a rod which runs inside the bacterium, shown here in blue. So that's part of the basal body. The basal body consists of a rod and a series of rings that anchor the flagellum to the cell wall and to the cytoplasmic membrane, as we see in the illustration. For example, in this gram-negative, the L ring stands for the lipopolysaccharide in the outer membrane. That anchors it to the outer membrane. The P ring anchors it to the peptidoglycan, and here's the rod running through, and that connects to the actual motor part down here at the bottom. So remember, unlike eukaryotic flagella, the bacterial flagellum has no internal fibrils and it doesn't flex. Instead, the basal body acts as a rotary motor, and that enables the flagellum to rotate and propel the bacterium through a liquid. Some of the proteins in the motor are called the stator, and that's a stationary part of a rotary motor. And that generates torque for rotation. And then there's a couple of rings, the MS and C ring, showing down here at the bottom in figure 1. And that's the actual rotor part of the motor, the part that actually rotates, in most cases either clockwise or counterclockwise. And energy for rotation comes from proton motive force. So proton motive force, again, is where hydrogen ions or proteins move across the membrane from greater to lesser hydrogen ion concentration. And as the protons are pumped across the membrane by the electron transport chain, the protons can re-enter via their gradient from greater to lesser concentration through these Mott proteins that make up the stator. And that in turn provides energy to rotate the rotary part of the motor. And this is a little movie that shows the assembly of the flagellum. So there's our bacterium, and now we go down with zoom vision and we see the motor being assembled in the cytoplasmic membrane here, the different parts of the motor. And that's part of the basal body. And then the rod is synthesized by proteins coming up through the motor. And then the hook is assembled to the rod. And now flagellin molecules synthesized in the cytoplasm are going to go up through the hollow rod and the hook, and they're eventually going to start polymerizing, producing a helical hollow tube. It's a little bit slow at the beginning of this, but it'll speed up in a little bit. So this is all part of the machinery getting ready to assemble the filament, the part that actually sticks out from the hook, this being the hook right here. So as flagellin comes up, it assembles, and you see the filament gets longer and longer as more flagellin is added to the tips. And of course, by this time, it's already rotating because of the motor inside, and that gets longer and longer until the fully formed flagella is functional. And this is in figure 4 is a transmission electron micrograph of E. coli 0157H7, where you see how long the flagella actually are. So a bacterial flagella can easily be 10 to 20 micrometers long, but they're only about 1/100th to 2/100ths of a micrometer in diameter. They're so thin we can't see them with a conventional microscope, but they are quite long. And there's a little self-check on structure. Now, flagella come in a number of arrangements, and these arrangements are distinct for specific organisms. The arrangements of the flagella that extend from the bacteria include monotrichous, lophotrichous, amphitrichous, and paratrichous. So let's take a look at some of these flagellar arrangements. Mono means one, so monotrichous is a single flagellum, usually at one pole. In this photomicrograph of Vibrio cholera, we see the Vibrio here with a single flagellum coming off of one pole. And here's an electron micrograph of a Vibrio where you can see how thin the flagellum is compared to the curved rod or Vibrio. An amphitrichous arrangement is a single flagellum at both ends of the organism. Some of the Spirillum species have amphitrichous arrangement where we see a single flagellum at both poles. Lophotrichous arrangement is two or more flagella at one or both poles. So this particular Spirillum here has a lophotrichous arrangement, and we see a bundle of flagella at each pole. In a paratrichous arrangement, flagella surround the entire surface of the bacterium, as we see in this Proteus here. And finally, axial filaments are internal flagella. These are found only in spirochetes. So these axial filaments can be composed of anywhere from 2 to over 100 of these axial filaments. And these axial filaments are sometimes known as endoflagella because they're actually located inside the cell rather than extending from the cell like the other flagella we looked at. And they extend from both ends of the bacterium, and they lie between the outer membrane of the cell wall and the peptidoglycan. And they often overlap in the center. So if we look at figure 6 in this illustration where part of the bacterium is broken away, part of the outer membrane is broken away, we see that it has the typical insertion structure down here at the end on the right, where we have the rod, the hook, and the filament coming out of the basal body. And then as the flagella elongate, they wrap around the spirals lying between the outer membrane and the peptidoglycan. And this is an electron micrograph of Leptospira on the right. You can see how thin the spirochetes are compared to other bacteria. And the little tiny humps you can almost see are where the endoflagella or axial filaments lie under the outer membrane. So this illustration kind of shows how these axial filaments lie between the outer membrane of the cell wall and the peptidoglycan layer. They wrap around the spiral. And it's thought that opposing directional motion of the axial filaments is what actually allows for the corkscrewing motion of spirochetes. So as we'll see in a video coming up in a little bit, the spirochetes have a typical corkscrewing type of motion, where it's thought that the rotation allows them to push back against their environment as the axial filaments rotate, and that propels the organism through liquids and through more viscous materials like mucus and cell membranes. And there's a little self-check you can do on that. Now, with regards to function, flagella, of course, are organelles of locomotion for most bacteria that have motility. And remember, the flagella rotate, as we mentioned, and it's driven by a proton gradient or by proton motive force. Now, most bacterial flagella can rotate both counterclockwise and clockwise. And this contributes to the bacterium's ability to change direction as it swims. And there's a protein switch in the molecular motor of the basal body that actually controls direction of rotation, clockwise or counterclockwise. Now, if we look at bacteria that are surrounded by flagella or have a paratrichous arrangement of flagella, counterclockwise rotation of the flagella causes them to form a single bundle. So all the bacteria kind of wind up wrapping around behind the bacterium from those surrounding the cell. And with counterclockwise rotation, this propels the bacterium in long, straight, or curved runs without a change in direction. So when it's rotating counterclockwise, the flagellum exhibits what's called a left-handed helix, which is hydrodynamically very stable. So during a run where we have long, straight motility, a typical run lasts about 1 second before the bacterium eventually stops. But during that 1 second, the bacterium can move 10 to 20 times its length before it stops. So it can move a great distance in a very short period of time on its molecular scale. Now, the stop occurs when some of the flagella in the bundle that will start rotating clockwise. And when some of the flagella start rotating clockwise, they disengage from the bundle, which messes up the hydrodynamics, and that triggers a tumbling motation where we see the bacterium just tumbling around. And with a clockwise rotation, the flagellum assumes a right-handed helix, and again, that's what leads to the tumbling motation. Now, a tumble only lasts about 1/10 of a second rather than a second like a run, and during that 1/10 of a second, there's no real progress made. But after the tumble, the direction of the bacterium, the next bacterial run is random. The bacteria don't really have any way to steer whatever way they're pointing. When they start rotating their flagella counterclockwise, again, that's the direction they move. And Brownian motion and fluid currents are constantly causing the bacteria to change direction during the tumbling. But eventually, one of those tumbles causes the bacterium to reorient in a good direction, and that results in, once again, having runs. So it's not like a bacterium moves from point A to point B from one end of under the microscope to the other, but rather it's a series of runs and tumbles and runs and tumbles and runs and tumbles. But the net movement of the bacterium, as we'll see when we get to taxis coming up shortly, is reorienting, is eventually taking the bacterium in a good direction. Now this video shows some E. coli swimming. And in this particular case with a conventional microscope, we can't see the flagella. E. coli has a paratrichous arrangement, but we can't see them. But what you can see nicely in this video are runs and tumbles. So if you take a look there, you see the bacteria doing nice little long runs, and then they start tumbling. So again, we see some of these tumbling rather nicely. So it's a series of runs and tumbles. Some of these are stuck to the slide and aren't moving, but again, you can see them not quite nicely tumbling and running. Now in this series of movies here from Howard Berg's lab, some of the flagella have been labeled with a fluorescent compound so that they fluoresce when hit with a laser, and you can actually see the flagella now on these bacteria. But what this is going to illustrate, again, this is E. coli that has paratrichous arrangement, but you'll notice that they form a continuous bundle behind the organism when they're running. But then as you watch them carefully, you'll see that some of the flagella slip out of that bundle, kind of like, it looks like the bacterium's doing the splits, and when it does that, that's what's going to trigger the stopping and the tumbling. So let's take a look at this movie. So there you see what looks like a single flagellum, but that's actually a bundle, and right there you just saw what looked like a little split. Let's watch that again. There you saw one with the, here, watch this, we see some of the back flagella come out of the bundle. And when you see that happening, that's when you get the stop and the tumble. Here's another movie kind of showing the same thing. So again, there's this continuous bundle, but some of them come out of the bundle. And when they do, that's what causes them to be hydrodynamically inefficient and stop and tumble. Now, there's another motion we sometimes see when bacteria are growing on a nutrient-rich solid medium, when they have paratrichous arrangements of flagella. And this is called swarming motility. What happens in swarming motility is the bacteria elongate, they get longer than normal, they synthesize many additional flagella, they secrete a wetting agent, and that allows them to actually move across a surface in a coordinated manner. So rather than going through a liquid, they're moving over a surface using this swarming motility. And again, the bacteria elongate and produce more flagella. And this is time-lapse of E. coli swarming. So you see they're eventually moving over the solid surface towards the right-hand side of the screen. Now, when bacteria have a polar flagellum, a single flagellum at one end, the flagella can also rotate both clockwise and counterclockwise, but in this case, when the flagellum rotates counterclockwise, it pushes the bacterium forward. When it switches and rotates clockwise, it pulls the bacterium backwards. So in this case, it's the clockwise or counterclockwise that determines whether it goes forward or backwards. And here's Pseudomonas, similar to the way we'll see it in lab under phase contrast microscopy. So again, you see runs and tumbles, but you see it changing direction depending on whether it's rotating clockwise or counterclockwise. But again, you see the runs, the tumbles. But in this case, the tumble is caused by the bacterium starting to change direction of the flagellar rotation rather than flagella flying out of a bundle, because these bacterium just have a single flagellum at one end. In spirillum that has a lophotrichous or an amphitrichous arrangement, the front bundle flips over towards the back bundle sticks out, or if it's a single flagellum, the front flagellum flips towards the back and the other flagellum trails behind. And when you see the spirillum moving here, you'll see it look kind of like a halo or a comet tail behind the bacterium. So there's the flagella that are trailing it, and you can tell the spiral shape as it swims, causing the spiral to kind of rotate, but there's the flagella sticking out the end there, propelling the bacterium. And there's a few bacteria of polar flagella that can only rotate clockwise, and that winds up pushing the bacterium forward. And when the bacterium stops, it's Brownian motion and fluid currents that reorient it, as we see in this Rhodobacter. But you can see nicely the flagellum does rotate in this. And there's a little self-check you can do. Which brings us to taxis, which is part of bacterial behavior. Now, about half of all known bacteria are motile, and motility serves to keep bacteria in an optimum environment. And this is done through a process called taxis. So we define taxis as a motile response to an environmental stimulus. So bacteria are showing behavior, they're responding to their environment. They can move towards a better environment or away from a more harmful environment. And different bacteria respond to different things. Virtually all bacteria with taxis have chemotaxis, and that's a modal response to a chemical stimulus. Bacteria that have photosynthesis that are modal have phototaxis, where they can move towards light. Bacteria also have osmotaxis, where they can move to be in a better osmotically balanced environment. They can respond to oxygen with aerotaxis, where obligate aerobes can swim towards oxygen, obligate anaerobes can swim away from oxygen, and they can also respond to temperature through thermotaxis. So let's look at chemotaxis. And chemotaxis is a response to a chemical gradient of either an attractant or a repellent in the bacterium's environment. So an attractant might be some essential nutrient they want to move towards. A repellent might be something harmful like acid that it wants to move away from. Now, if the environment lacks a gradient, such as often happens when you just have them growing in a test tube, where there isn't a gradient of more attractant here or more repellent here, bacterial motion is randomly because it's looking for a gradient. So in a lot of those movies we saw, we're looking at bacteria where there was no gradient, and that's why you saw it changing direction and moving. It was looking for a gradient. But if an environment has a gradient of an attractant or a repellent, the net movement of the bacteria is towards the attractant, the good substance, or away from the repellent. Again, it's not a continuous movement, there's runs and tumbles, but the net movement is towards an attractant or away from a repellent. And as we mentioned with Paratrichus bacteria, this change in direction is triggered by tumbling from some of the flagella coming out of the bundle and rotating clockwise, causing it to tumble and change direction. And with in this movie here, we can see some tethered E. coli. In this case, the flagella have been attached to the microscope slide, but not the bacterium. So the flagella, although the flagella are rotating, the flagella are attached at their distal end. And so we actually see the E. coli rotating on the end of the flagella. And you can see it change direction and rotate clockwise and counterclockwise. So there we see the E. coli rotating because the flagella are attached to the slide. You see it change direction there, clockwise, then counterclockwise. And that was some of the first evidence that showed that bacterial flagella do in fact rotate. So chemotaxis is regulated by chemoreceptors located either in the cytoplasmic or the periplasm of the bacterium. And these chemoreceptors bind an attractant or a repellent. And what happens in most cases is that as the attractant or the repellent binds, this can lead to either methylation or demethylation of some chemotaxis proteins. And it's this methylation or demethylation, adding or removing a methyl group, that eventually triggers either a counterclockwise or clockwise rotation of the flagellum. So an increasing concentration of an attractant or a decreasing concentration of a repellent, both conditions that would be beneficial, causes less tumbling and longer runs, and that's why the net movement is towards the good environment. And again, a decreasing concentration of attractant going away from something good, or an increasing concentration of repellent going towards something bad, both these are harmful conditions. This causes tumbling and a greater chance of reorienting in the right direction. So the net movement is towards an optimum environment. Now, in several places in Unit 1, we've talked about the initiation of body defenses. This will be the last time we're reviewing these terms in Unit 1. But again, by reviewing them quite a few times in Unit 1, hopefully by the time we get to Unit 5 on innate immunity and Unit 6 on adaptive immunity, you have these terms down fairly well, which will make our task easier at the end of the course when we get into the body's immune responses. So remember, innate immunity is antigen non-specific. Our body uses this immediately or within several hours after exposure to a microbe. And this is the immunity we're born with and the body's initial response to try to eliminate microbes and prevent infection. Whereas adaptive immunity is antigen specific. This takes several days to become protective and is designed to remove a specific antigen. And this is the immunity we develop throughout our life. So to initiate innate immunity, again, we look for molecules that microbes have that human cells don't have, but molecules that many microbes share, so we can recognize whole groups of bacteria with innate immunity. And remember, what we look for are pathogen-associated molecular patterns, or PAMPs, molecular shapes on the microorganism or pathogen in this case. And in case of bacterial flagella, the protein flagellin is a PAMP. It's a pathogen-associated molecular pattern. When flagellin binds to pattern recognition receptors on defense cells, that triggers innate immune defenses like inflammation, fever, phagocytosis, just like cell wall PAMPs do. Then to initiate adaptive immunity, proteins associated with the bacterial flagella function as antigens, and they initiate adaptive immunity. So again, an antigen is a molecular shape that reacts with antibody molecules and with antigen receptors on lymphocytes. So we recognize the antigen as foreign because they fit antigen receptors on our B and T lymphocytes, the two cells we use to carry out adaptive immunity. But again, we don't recognize the whole antigen as foreign. We recognize little bits of it that have unique shapes. So the actual portion or fragment of that antigen that actually binds to an antibody molecule or binds to a receptor on a B or T lymphocyte, again, is called an epitope. And if that's a protein antigen, hundreds of amino acids, remember there are certain amino acids at different locations in different orders that have certain shapes. And so these little mini shapes stick out of the antigen. So each one of these is an epitope. A few amino acids, typically between 5 and 15 amino acids, sticking out of the antigen that we recognize as foreign. And that's what eventually the antibody reacts with. But each of these antibodies tailor-made to fit that one epitope. And these are also the epitopes that would bind to receptors on B and T lymphocytes to initiate adaptive immunity. So again, protein antigens, like in figure 8a, have many different epitopes on them, each stimulating an adaptive immune response. Whereas if the antigen is a polysaccharide, then 3 or 4 sugars branching off the polysaccharide acts as an epitope, as we see in figure 8b. But again, because there's only 1 or 2 sugars in a typical polysaccharide, although there's many epitopes along that polysaccharide, they're all basically the same epitope. And remember, we have 2 branches of adaptive immunity, humoral immunity and cell-mediated immunity. So humoral immunity is the production of antibody molecules in response to an antigen. That's carried out by B lymphocytes. And again, as we've seen, these can neutralize microorganisms or their toxins in a variety of ways. So when antibodies are made against flagellar antigens on bacterial flagella, they can actually block motility or arrest motility by binding to the locomotor organelles. And by stopping motility, that might arrest the organism's ability to spread or to respond to the environment. And of course, antibodies made against flagellar antigens can also act as opsonizing antigens or opsonizing antibodies to bind to the flagella and then stick the bacterium to a phagocyte. Whereas during cell-mediated immunity, this is the production of cytotoxic T lymphocytes, activated macrophages, activated NK cells, and cytokines in response to an antigen. And this is carried out by T lymphocytes. And these defense cells help to remove infected cells and cancer cells that display foreign epitopes. Now let's look at the significance of motility to bacterial pathogenicity, that is, the ability to colonize and cause disease. First of all, motility and chemotaxis probably help some intestinal pathogens to move through the mucus layer secreted by the mucous membranes, so the bacteria can eventually reach epithelial cells of the mucous membranes, where they can subsequently attach with cell wall adhesins or with pili or with a glycocalyx. So many bacteria that are able to colonize the mucous membranes of the bladder and the intestines are motile. And again, they probably help the organism to get through that protective mucus layer that's trying to flush them away and eventually adhere to epithelial cells where they can colonize and resist flushing. And not all the mucus has the same viscosity, so the motility probably helps them to move through mucus in places where it's a little less viscous or a little more watery. And this animation shows you a bacteria with a polar flagellum. And this is the mucus layer that surrounds the mucous membranes, let's say, of the intestines. And various nutrients down here from these epithelial cells form an attractant. And as they diffuse away, we get a gradient where we have increasing attractant as we go down towards the mucous membrane. So the bacterium can use its motility to swim through the mucus towards increasing concentration of attractant till it makes contact with the epithelial cells of the mucous membranes. So there we see it moving towards increasing attractant until it gets down to the epithelial cells. And then again, it can use cell wall adhesins or glycocalyx or pili, which we'll take up in another SoftChalk lesson, to attach. Now, motility is also very important for pathogenic spirochetes, bacterium like Treponema pallidum that causes syphilis, Leptospira that causes leptospirosis, and Borrelia burgdorferi, which causes Lyme disease. So motility and chemotaxis enable spirochetes to actually move through viscous environments and even penetrate cell membranes, viscosity equivalent to that of a cell membrane. In fact, my doctoral work long ago was with Leptospira, where I helped show that spirochetes are hydrodynamically designed to move through more viscous environments. An environment that would normally slow down and stop bacteria with external flagella actually swim better in a more viscous environment. They can move faster. And this is because of their thinness. The spirochetes are some of the thinnest of the bacteria. So by being very thin, there's less body of the bacterium at the tips to block their forward motion. And you don't have the damping action of the viscosity or the sliminess preventing the flagella from rotating, because the flagella lie inside the cell between the outer membrane and the peptidoglycan, the axial filaments. So because of their thinness and their internal flagella and their motility, spirochetes are more readily able to penetrate host mucous membranes and skin abrasions, so sometimes helps in entering the body. But also what we see in a number of these spirochetes is that the disease comes in stages. And this is where motility and invasins and chemotaxis enable the spirochetes to penetrate not only deeper into the tissue from the initial entry site, but then they can actually enter the lymphatics and the bloodstream and disseminate to other sites in the body. And we see that in this animation here. So this is a capillary right here, which is made up of a single layer of endothelial cells. And of course, the blood cells and the plasma are flowing through here. Now, what the spirochetes can do, the pathogenic spirochetes, is their tips have an adhesin that kind of allows them to adhere to the surface of the cells at their tip. And the internal flagella cause them to have a corkscrewing motion. They also secrete some invasins that can soften up the membrane of the cell. So the combination of the corkscrewing motility and the invasins and attaching just at the tip causes them to actually corkscrew through the membranes of the cells, the endothelial cells, and in this case, enter a blood vessel. So they can use the motility to actually drill through cells and enter the lymphatics or the blood, or they can pass between the endothelial cells and enter the lymphatics or the blood. So we see the one on top is going to drill its way through an endothelial cell and enter. The one on the bottom is passing between the endothelial cells, and the blood then carries them elsewhere in the body, where the same motility allows them to adhere on the inside and pass out again and set up a new infection at another body site. So this corkscrewing motility of the spirochetes does play a role in some of the pathogenic spirochetes disseminating and actually coming in stages. And here we see a spirochete under dark field microscopy, where you can see the tip and the corkscrewing motility. This is actually an ad for something called Cytoviva, which allows the spirochetes to jump out a little bit more under dark field where you can see them better, but in either case you'll see the corkscrewing motility here. So there you see the tip, and as the axial filaments change direction of rotation, they can either make it go forward or backwards. But again, you see the leading end doing its little corkscrewing motility. And here it is after Cytoviva, so you can actually see the rotation quite nicely in the tips, allowing it to propel itself through the medium. And here's a really nice electron micrograph of the syphilis spirochete, Treponema pallidum, invading a host cell. So here is a Treponema pallidum, a spirochete that's drilling its way through a host cell or into a host cell. Here's one that's almost all the way in. So again, by being able to attach just by the tips with an adhesin, like kind of the starting point for a drill bit that's rotating, and then the corkscrewing motility and the invasins that are kind of softening up the plasma membrane, these can enter through cells or enter into cells with their motility. And there's a self-check on pathogenicity. And at the end of the unit, of course, we have our self-quiz on the whole SoftChalk lesson on bacterial flagella.